Evolution of Metazoan Morphological Disparity by Bradley, Deline, and others discusses the idea that the Cambrian explosion is over-dramatized and phenotypic evolution does not occur in a continuous line but rather in discrete jumps and starts. Before we get into further discussion about the paper, we need to define some important things in the paper, one of which is the Cambrian explosion. This refers to an increase in the biodiversity of multicellular organisms at the start of the Cambrian period 540 million years ago. Another main point is that evolution occurs in starts and fits, and they discuss how major changes in the animal form after the Cambrian align with ecological transitions like the exploration of new land. There are some complex words in the title of this paper that are vital to understanding the research as a whole. Metazoan refers to a zoological group comprising the multicellular animals that develop from an embryo. Morphological spaces are spatial representations of the difference between organismal phenotypes. For their purpose, disparity means the difference between phyla and characteristics of the morphospace. The paper makes connections to major themes in biology, including evolution, Earth history, and history of life on Earth. Something included throughout the paper is phylogenetic trees, which are diagrams showing the evolutionary connections of a group of organisms deduced from a common ancestral form. Some other terms used in the paper are fossil record, which is a term used by paleontologists that refer to a total number of fossils that have been discovered, and adaptive radiations, which refers to the diversification of a group of organisms into forms in separate niches. The term phylum or phyla refers to taxonomic classification groups which rank below a kingdom and above a class.
In the past, the Cambrian explosion has been thought to be the epitome of evolutionary changes. This is because collected evidence has shown to us that many phenological changes occurred for metazoans during that period. However, this evidence is limited to fossilizable characteristics, as soft tissue is not as well preserved as fossil evidence. Thus, the explosion of the Cambrian has been exaggerated and shows the change in fossil characteristics rather than overall phenotypic disparity. This is furthered even more through morphospace mapping, which the researchers use to demonstrate that the disparity of characteristics of certain species occur progressively, rather than the sudden appearance of evolutionary changes. Furthermore, the researchers noted that there has not been enough time in Earth's evolutionary history for all of the possible metazoan phenotype characteristics to be explored, possibly resulting in the clumping or discrete nature of the metazoan morphospace occupation. They also believe that many metazoan species had the potential to evolve more characteristics, but it is now impossible to because the appearance of these characteristics is dependent on the existence of a combination of characteristics that had to exist earlier in their phylogenetic history. The second major aspect is how morphological distances between phyla mirror disparity in complexity, body size, and species-level diversity across the animal kingdom. Analysis of maximal initial disparity of clades indicate that arthropod, chordate, and mollusk lineages expanded the limits of the morphospace from marine to terrestrial throughout the Phanerozoic. There is a strong correlation between increasing morphological disparity, genome size, and microRNA range, but no correlation to protein domain diversity. In other words, the evolution of gene regulation and alterations to microRNA shaped metazoan disparity, and the transition from water to land supports the Cambrian expansion wasn't a non-uniformitarian change. The uniformitarian change observed is due to the similarity of intrinsic processes, which generate underlying genetic and developmental variation. The results also suggest that animal evolutionary history follows a high frequency of small changes and a low frequency of large changes in the context of internal genetic variation and external environmental change. So, animal evolution should actually focus on debating the chronological location of magnitude changes. Future research should focus on the nature of phenotypic evolution, its relation to molecular evolution, and developing phylogenetic methods to find a more accurate tempo of metazoan diversification.
Overall, the paper reveals some major misconceptions about how the world and scientists perceive evolutionary history. First, it highlights how most phenotypic disparity between phyla is not attributed to a single event, namely the Cambrian explosion, meaning the Cambrian was more like the Cambrian. And second, the paper points out that evolution did not occur as Darwin once thought, a gradual change in characteristics. And these findings shed new light on life's history and how scientists view the timescale for phylogenetic evolution. The findings help us understand how and when certain phyla gain their defining characteristics, and more importantly, that gene regulation was key in allowing for the evolution of metazoan organisms.